Namaskar, I am Professor Lalit Pandey, PIE of Biointerface and Environmental Engineering Laboratory at Department of Biosciences and Bioengineering, Indian Institute of Technology, Guwahati. My research work in the area of biochemical and biointerface engineering and applied research is related to healthcare and environmental applications. The major challenge in the bioremediation of spilled crude oil sites are mainly three. First one is potential strains, second one is cell surface hydrophobicity, and third one is availability as well as toxicity of hydrophobic oil substrate. The hydrophobicity of the oil significantly hinders its biodegradation. This causes limited access to nutrients, reduced bioavailability, and slow degradation rates. Further, the cell surface is hydrophilic, which results in poor integration with hydrophobic substrates. In this regard, first we have isolated different indigenous bacterial strains from various sites and enriched them for particular phenotypes. For example, degradation of oils. The hydrocarbon degraders are found to produce surface active molecule that is biosurfactant. The biosurfactants improvise substrate oil availability through emulsification, which in turn improve the solubility as well as bioavailability of the oil, facilitating the faster degradation. Finally, the biosurfactants also enhances the cell surface hydrophobicity. So in this way, biosurfactants are able to address all three concerns. Further, this strategy has enhanced the oil degradation and its efficiency by many folds. And next, we have developed a consortium to improve the overall degradation. So biosurfactants have been produced at shake flask as well as at bioreactor levels using different substrates. The maximum yield is found to be about 12 gram per liter. The interfacial properties of the biosurfactant systems are found to be very impressive, with lower value of critical micellar concentration and surface tension. These surface active molecules have been explored for a wide range of environmental and biomedical applications. Some of the potential applications of biosurfactants include microbial enhanced oil recovery, where we are improving the oil extraction efficiency from underground reservoirs, bioremediation for the cleaning up of oil spills in oceans and soil sites, cosmetics and pharmaceuticals, where we are replacing synthetic additives in skin care and drug formulations, also excipients for biopharmaceuticals. On the same line, in this study, a potential strain from Pani Tenga, a traditional oil-rich fermented food from Assam, was isolated and identified as Bacillus subtilis SMP2. This strain was selected based on the oil degradation phenotypes and has the unique ability to produce biosurfactants. The process parameters were optimized to maximize the biosurfactant production. In the lab scale prototype, almost complete degradation of 10 gram per liter of crude oil has been achieved. Further, my research team is exploring the integration of this process with a hydrophobic bioabsorbent system. This approach aims to enhance the oil adsorption rate before facilitating the bioremediation using the bacterial strains. This innovative method holds a promise for more efficient and sustainable management of oil spill waste in marine environment. This work is supported by Department of Biotechnology, Government of India and the Ministry of Education, Government of India. Thank you.
Hello, my name is Smriti Sonbhadra. I am a PhD scholar of IIT Guwahati from the Department of Biosciences and Bioengineering. I work under the supervision of Professor Lalit Pandey sir. So our research group basically works on the production of biosurfactant. Biosurfactant are the amphiphilic molecules. Basically they have both hydrophilic and hydrophobic groups together. So we produce biosurfactants from bacteria basically. Since biosurfactant name itself reflects, it is a biological source product. So we are producing biosurfactants from bacteria, and these bacteria we are isolating from different sources. For example, from seawater, from some oil reservoirs. Also we have collected some samples and we have isolated bacteria from it. And the current work basically, I have isolated a bacteria from a fermented food product whose name is pani tenga. It is an Assamese fermented food product. So we have isolated a bacteria from it, and we have tried to see that how much biosurfactant that bacteria can produce. And for that we have performed several screening tests. One of the tests is oil displacement test. In that test we see the ability of that bacteria that produces biosurfactant to see whether the oil which is present over the surface is being displaced by that biosurfactant or not. So if the bacteria is producing biosurfactant, the oil will be displaced from the surface. And this property is further being utilized in various other sectors, like in oil cleanups. Oil cleanups is basically oil remediation practice. For example, in sea waters there are several oil spills accidents that have occurred in past. So our aim is to use those oil-eating bacteria with the help of biosurfactants to clean those oil-contaminated sites. And also we use those biosurfactants produced by bacteria in other applications, like for detergents application or for the production of antibiotics. So these are the other different areas that we are exploring the produced biosurfactants from the different isolated bacteria. So we are isolating bacteria and we are trying to produce maximum biosurfactant from that bacteria by giving cheap carbon sources like waste carbon sources to make the overall process economical, like molasses, cornstarch liquor and glycerol. So these are waste cooking oil is also one of the carbon sources, cheap carbon sources that we have used, our research group has used. So the ultimate aim is to isolate bacteria, potential bacteria, produce biosurfactant and use that biosurfactant for various applications.